Hey guys, good morning. So this one is about the continuous about this gene expression. So let me do this one here. Share screen. That's this one here. All right, share screen. All right, so you can see this one here. So last talk is about the gene expression, and this talk is about how are those steps to regulate the gene expression. Um, so what's the gene expression step of the regulation? There are several steps that we can regulate our gene expression. First is to control whether to transcribe or not. Gene expression has two steps, right? Major steps are two: transcription and the translation. Transcription is from the DNA to RNA. Translation is from the RNA to protein. Okay, so you know that. Now, so in these two steps, there are several ways that we can regulate it. The first is to work on the transcription. So to control whether to transcribe it or not. Okay, that's a very important one to make it easier to transcribe or make it more difficult to get transcribed so the gene can be expressed or not. So the very first step is to control the transcription. And the second one is the RNA process. Once we generate an RNA, DNA to RNA, this RNA can be further processed. One important one is called the splicing. We will talk about that. So that can make these RNA to become different variety. Then we have the protein, right? So transcription, then RNA to protein is translation. After translation, we can have several, during that after translation, we can have several protein process to post-translational modification. So that's all this. Let me show you this one here. So this summarizes several steps that we can do. We can control the gene expression. So here is the DNA. DNA has replication, right? Cells can divide. And then once the DNA is ready to be transcribed, it will go through the transcription to produce the RNA. And the RNA will move out of the nucleus and will be translated to protein. And in our earlier talk, last one, we talked about how this is produced. So we have the idea, the standard procedure, how this is complete. Sorry, let me get something to drink. All right, so and this one is about how do we regulate. So here you can see that several things can be done. We can do the chromatin remodeling. We can transcribe that can affect the transcription to make it transcribable or not. We can have alternative splicing that is working on the RNA process. Then we can have through this translation, we can have microRNA to block the protein synthesis. So even we have the RNA, we can still have one step, very last one, not very last one, but one step to prevent the translation. That's the microRNA. So now you learn the one new word, that's microRNA, right? That's a type of the RNA, but pretty short. And its function is to conduct the break, to say, no, don't do it. Come over here and stop the translation. After we have the protein, we can have several processes. And so protein folding to fold it into the correct form. We can make a polypeptide shortened to truncate it, to make it short. We can add sugar to it, so make it glycoprotein in a way. to add glucose to it, some decoration. It's like you made bread, right? You made some sugar to it, some decoration. And then we can also have a polypeptide aggregate that this peptide can be aggregated. And then we can form different proteins. So this is not just the stop or start, kind of like brake or accelerator. It's also a way to make our products to be much more diverse. So from the same DNA gene, we can have a different kind of products, different kind of protein in the end. We can control the speed, we can control the amount, we can control different variety. So that's the topic that we are going to talk about today. So basically we will pick the very important one here and to show you how our cells regulate this gene expression. So before this talk, you must take the previous talk, the typical standard gene expression process. Because if you don't know that, you cannot know this one, because that's the one, that lay the foundation that you know how gene expression is worked. So at least you need to know what's the transcription, what's the translation, so you need to know this part. And we are going to talk about this part today. So before we jump into that regulation, just a quick summary of what is the thing that we are dealing with today. That is the DNA, right? So the initial material that we are going to process and regulate is the DNA. So DNA is it's a nucleic acid that is a polymer, right? And that that contains the genetic information. And so in our cell, we have the chromosome. See, here is a chromosome. This is the figure showing you that, in fact, two chromosomes are duplicated, connected with the centromere. So when you see like this kind of butterfly shape, it means that this is in the cell division process, probably in the S phase, right? That's after the cell. That was the phase that chromosome duplicated to produce sister chromatids. If you forgot it, go to this earlier talk. When we talk about the cell physiology, when we talk about the cell division, we talk about that. So that's that. Otherwise, a single chromosome will be only one, not two that linked with the centromere. All right, so that's that. That's chromosome. If it's single one, it's called chromatin. All right. So this chromosome, single one is also called a chromosome. Sorry, and this chromosome can be, and then this chromosome is a packed one that we see in the nucleus. And then here, extended, if we unfold it, this one is called a chromatin though. So the fiber, this fiber, if we further fold it, it will be chromosome. If we unfold it, but it's still very highly condensed folded. This is called a chromatin fiber. Now this fiber, this is composed of the two important components, that is the DNA and the protein. So the chromosome is a package of DNA and protein. The DNA is a DNA that we know. It's a polymer of the nucleotide. It contains genetic code GCAT, right? And the protein, what's the protein? This protein is called histone. So histone is a protein. The function of the histone is to provide these spool to make this fiber to wrap around it. So that's the histone. So the function of histone is to provide that central piece. So the DNA can wrap around it and make it condensed. So that's the histone. Typically, there are 8 histone work together to form a octone. We will know that later. So 8 histone together and DNA will wrap around it. These little bits serve as the unit structure, the basic unit structure of the DNA packaging. This bit is called the nucleosome. So just terminology. When people say this, you know what it means. Chromosome is not just the DNA. Chromosome is the DNA and the histone. And nucleosome is the little bit, the unit structure of the DNA packaging of the chromosome. This one shows you the transition from the DNA into chromosome. Sometimes we hear chromatin, like very beginning, we say it's called the chromatin here, chromatin remodeling. So chromatin basically is chromosome. It's just different level of packaging. So here, where is it? Here, chromatin fiber. So this is chromatin fiber. And only that this, the difference between chromatin and chromosome is basically is how condensed it is packed. These two are the same in the way that they are DNA package, and it can include the DNA and the histone. So this is already there. It's just from the chromatin into the chromosome is further condensed. Usually, we don't see this chromosome structure unless the cell is in the cell division, cell divided process. Only when we see the chromosome is duplicated, then we see this kind of structure. And before that, this chromatin need to be packed in order to become ready to be split away, right? Because after this process, the next step is the mitosis, right? So these two chromatin will be separate away, will be pulled away. And so we need to pack it further to become the chromosomes. And that way, when they pull it away, it won't be damaged. If it's loosened like this one, if we pull them apart, you're gonna cause some kind of the damage. So that is that. It's the difference between chromatin and chromosome. It's just how condensed they are. And typically when we do chromosome imaging, this type of technique is called karyotype. A karyotype, we will see layout of this chromosome, right? Because this is not freely, it's not entirely freely, but less condensed structure. So this one is one chromosome. So this figure showing you that before the chromosome duplication, we have one copy from the mother, one copy from the father, and one copy from the mother, right? So we have 23 pairs of chromosomes. These two chromosomes do not connect with the centromere, though. Centromere, they don't. Only those sister chromatins, when they are duplicated, they are connected. Otherwise, they are separated. One from dad, one from mom. We can pair them together. When we stem them, we can copy and we can chop it and then move it. Okay, these two are together, and so we can show we have two copies from the same chromosome. And so this is single chromosome, like this one here, single chromosome. And during the S phase, when chromosomes are replicated, we have two. Now these two are connected with the centromere. So this one showing you that this is a chromosome, and just like this one here, and then it duplicate. Then this duplicate 2 will connect it with the centromere. And so this one is called chromosome, have two sister chromatids, these two sister chromatids. Before we pull them apart, we need to condense them further to make it chromosome. But still, this is called chromosome. So one chromosome, this is one chromosome, this is one chromosome, two sister chromatids, one chromosome, two sister chromatids, and then we separate them apart to call them two chromosomes, one chromatid each. So just some terminology. You know the level, chromatin is less contents, chromatin. Chromatid, TID, is two sister chromatids. This represents the sister chromatid, the duplicate, the replicated chromosome. And the chromosome is one that highly condensed. So that's that. All right, we have some background here. Now, let's see how do we do chromatin remodeling. Chromatin remodeling has two remodeling. One is called histone modification. So that work on this histone. The other one is DNA modification. So that is the one to work on this DNA. So we have two modifications. Once we have this chromosome, we can do several things. Cells do several things. One is to modify this histone. The other one is to modify this DNA. So let's see how it's done. So before we talk about this histone remodeling or modification, histone, let's get a closer look at the histone. So histone, we have 8 histone, as I showed you earlier, 8 histone together to form a protein complex. This complex is called the octamer. Octam basically means 8, right? That's very interesting, right? Because say October is actually, it's not sequenced in 8, but the original idea is 8, but we add 2 other months before. So October become the 10th one if we consider January the 1st one. All right, now, so November is November the 9th one. November, in fact, is 11th one, right? October, okay. All right, so this is the histone. Histone has 8. These 8 histone are a pair of 2 copies of each of these 4 histone proteins. So you know there are 4 major basic histone proteins: H3, H4, so histone 3, histone 4, and H2B and H2A. So H2A, H2B, H3, H4. So there must be some history that we first identify H1, 2, and realize that they are exactly the same as maybe 3, 4, right? So in the end, we have confirmed that there are 4 types of histone. These 4 are different. They are H2A, H2B, H3, and H4. And even though we have total of 8, these total of 8 are like a same copy of these 4. So we have these 4, and that's one thing. So one thing is that these 8 have come from these 4 histone. They together, and then DNA, you don't need to worry about that 147 base pair. DNA, that number you don't need to know, but you need to know that this one DNA wrap around, and this is called what? This whole like structure is called a nucleosome, right? So that's right here. That is the nucleosome. So that little basic unit here is called a nucleosome. I can write that. Is that this one? So that's the nucleosome right here. Okay. So that's that. Now, another thing about this nucleosome is that, another thing about this histone is that this histone contains tails. So they have tails. So this is just another picture showing that DNA wrapped on histone, and this is nucleosome to form the chromosome. These chromatin fibers and form this chromosome. Now, histone, this protein has tail. This tail extend out. So it's not in here, they extend out. So this is the tail. This tail can protruding from this nucleosome, and it's a place to be modified, meaning that they provide the way that you can do some decoration to it. So I want to show you this picture. So you can see that there are 8 histone, right? 8 histone here. We have four type of histone. These 4 type of histone, say H3, H4, H2A, and H2B. Each histone has a tail. So you can see this is the tail. Each letter represent an amino acid. And to this N-terminal. And this amino acids, from any N-terminal, we have the number to it. So 5K, this is one amino acid located, the position 5. P10, that's the position 10. We have the amino acid P, 15, 20, etc. So we have the position number. We also have the amino acid. So we are very clear that what's composed in this tail. Okay, we have 4 from 4 different histones. Total, we will have 8 tails, right? Because we have 8 histones. But we use this 4 as representation that, say, H4, we have 2 H4. So each one will have this tail. All right. And we can see that we can do several decoration to it. So this is basic structure. And we can do 3 type of modification. We can add a phosphate to it. We can put a methyl group to it. We can put an acetyl group to it. Methyl group, acetyl group, methyl group, phosphate group. So we have 3 types. A lot of these are decorating to H3. H3 has the majority, has the major histone tails to get modified. We also have a lot position for H4. These 2 are very important for methylation, methyl group to attach to it. And we also have acetyl group to attach to this H2A and H2B, H3, H4, right? And we also have some location for the phosphorylation, phosphate attached to it. So that's that. We have a lot of opportunity that we can do histone modification. So go back to this one, that's a tail. And if we zoom into a tail, you can see that a lot of this site to get modified. So this one summarizes what are those histone modification. We have the acetylation to add acetyl group right here, acetyl group to lysine. Lysine is the amino acid. on specific histone. So that's one. Acetylation. So three types of the modification: acetylation, methylation, phosphorylation. Acetylation add acetyl group to, and acetyl group can only attach. The most common one is to attach to the amino acid lysine. Methylation add methyl group, C3 to lysine. And methylation, one thing unique is that it attached to H3 and H4, the most common one. And the phosphorylation add add phosphate group to the histone tail. So that's three. Done. You know it. So just figure showing you that how they are attached to it. Histone acetylation attached to. So this is the histone. It has 8 tails, right? And a lot of his acetylation can add to several of these tails. Now, what is the function? What's the meaning of acetylation? The function of acetylation is to help to turn on or turn off the gene expression, meaning that if acetylation attached to it, this group, this segment of gene will be expressed, has higher chance to get expressed. If there's no acetyl group attached to it, this is a process to make it silenced. Yes. So that's that. So that's what you need to know. Will acetylation cause gene expression? Yes. Will acetylation cause gene expression? No. Deacetylation causes genes to turn off. So that's that. So basic idea is that the DNA carries negative charge. Acetylation would mute or naturalize it, provide a positive, so there's less to the histone to neutralize histone positive charge. So there's less attraction from the histone and the DNA. So the DNA will be less attached to the histone. So it will become more accessible by transcription factor, right? And RNA polymers. And those two will bind on it and cause the gene expression, cause the transcription. So that's that. This is the picture from the textbook. So if you look at that, it's kind of complicated, right? So the basic idea is that that's a quiz question that will acetylation on the histone cause gene turn on or turn off? The answer is turn on, okay? So that's the only thing you need to know. This one showing you how it's complete. Basically that we have this enzyme called the acetylase combined with the enzosome and enhancosome. So this one basically attaches the enhancer of the sequence in the gene. We didn't really talk about that, but you know the idea. And then they will add the small protein complex, and that will cause the acetyl group to attach to the histone tail. Without that, it will become turned off. With that, it will turn on. And this is this large protein, including transcription factor and RNA polymers, will come here to this promoter sequence, the Tata box, and it will start to conduct this transcription. So that's the acetylation. Easy, right? Turn on or turn off. Now the methylation. Methylation, what do you need to know? You need to know that it adds to the methyl group CH3 to the histone tail. What you really need to know is that its majority is to add on H3 and H4 tail. And they are not like a distinct difference between whether it must turn on or turn off the gene. It will turn on in some situation, and it can be turned off in some situation. So that's so the quick question would be, where are these histone methylation the most commonly observed in the histone? It's on the histone 3 and histone 4, H3 and H4. They are located, they are decorating this methyl group to the lysine and arginine. So here showing you that if it's X on the H3K4, so it's where H3, this is H3, 1, 2, 3, 4, K4 right here, right? This is 5, K4. So if this one, then it's turned on. It's turned on. H3K48, H3, 48 right here. This one, K48 is turned on. But don't memorize it. You have the idea. That's good enough. Okay, because there are so many methylation sites, and there's no way that we can memorize all this unless we do this, every day. Otherwise, we have the idea that there are many methylation sites, and it could be turning on or turning off. So the majority is on the H3 and H4 histolation on the H3 and H4. So you see that from the N-terminal back to the core of this protein, we have these long tails, and they serve as the site to be modified. And that can affect the gene expression. So we regulate it. All right, so that's that. In addition to the histone modification, as I mentioned before, in addition to the histone modification, and that way we can modify the chromatin, right? It can become less expressible or become more expressible. We can turn on, turn off the gene expression. In addition to the histone modification, we can also have the DNA modification. So the next step, and the histone modification has 3: acetylation, methylation, and phosphorylation. Easy, right? So these 3. DNA modification, only 1: methylation. So that's it here. DNA methylation. SHIFT3H2 what? SHIFT3 is add to cytosine. Okay. DNA has 4 different nitrogenous base. GC80, guanine, cytosine, thymine, adenine, right? These 4. And methylation is always on cytosine. Okay, so that's that. So this is the cytosine. Cytosine will add the CH3 to it. It's on this 5 position. This DNA, this nitrogenous base, for the cytosine, thymine, uracil, they have similar structure here. This is called pyrimidine ring. This ring is called pyrimidine ring. Very similar, some variety. So this is the cytosine, this is the thymine, and this is the uracil. One thing that I want to get your attention is that So this is the cytosine that this 5 position can be modified, can add a methyl group to it. Now, if we put a methyl group to it, it will be a CH3 here, right? And then we must know that cytosine, this NH2 group, has the tendency to become, because this bonding is not very strong. This can be called deaminated. This NH2 can become leaving or coming back. If there is no modification, if this NH2 leave, this will become something can be correct, because if this leaving, it will be attached to an O. So this one will be viewed as an uracil, and in the DNA, there's no uracil. So this can be correct back to the cytosine, because cytosine is connect with the guanine. So a pair GC, if there is any deamination, it will become GU, uracil here. And that can be easily fixed back to GC, because GC, AT, we know the rule, the cell know the rule too. So it will know, you see, if the cell see this one is a GU, it will know something wrong with this U. There's no supposed to be a U here. So it can fix it. And so that we can maintain, even though there's potential to be deamination, it can be fixed back to GC. Now, if this cytosine attached to a CH3, then with a CH3 here, following a deamination, this NH2 removed to become the O, then we will see it as a thymine. This cytosine, methylated cytosine plus a deamination will produce a thymine. Then we will have a GC become a GT. GT is not right, because GT is not does not meet into the best principle, right? GT is not right. Then when the cell see use this one. When a cell see these, when the cell see these become GT, right? Because cytosine following, methylation, CH3 to it, and then removing this O, it become a T, right? Like this one here. Like this one here. So it will be GT. GT is not right. It's either GC or it's either AT, right? These two are correct. These two are correct. This is not right. So the cell will try to fix it. It can fix it back to GC, or it can fix it back to GT, AT, but not GT. GT is not correct. It's not, doesn't fit this pair principle. And when it fixed to GC, then nothing is changed, because it was supposed to be GC. But if fixed to the AT, then we will have a mutation, because the original call is supposed to be a C here, but we make it, this one become a T, G, and then it becomes a TA. So a GC become an AT, and then we have the mutation. So that's the situation that DNA methylation has a tendency to cause mutation. Clear all drawing. Yeah. All right, let's go back to this one here. So that's this, let me see. So that's what we have here. So this is the cytosine methylation, and if there is no deamination, this one will be a cytosine here, but deamination happens all the time. So that's this one here. DNA methylation has a tendency to become a mutation. It's because that methylation on the cytosine to form a 5, position in 5, methyl cytosine, and then will be a very similar like the 5 position in the thymine. And there is spontaneous deamination. So I probably should. Spontaneous deamination that remove this NH group, NH2 group, replace it as an O. Then we will have the thymine. So that's the situation. So here in the beginning, we have this CG pair, like this one here, GC-CG pair. And the C become T through the methylation and deamination. Now TG is not correct. TG need to be fixed. TG can fix back to the CG or to fix it to become a TA. And if it happens to be the TA, then it will lead to a mutation. And that leads to a consequence that in our genome, the number of CG is actually is much lower than what we expected. That is because that C is a site to receive this methylation. And in fact, we have a lot of DNA methylation in our genome. And that methylation has tendency change it to become a TA. And so through all this history, those mutations become part of us that we are basically can survive and become okay with that mutation. But if you probably looking back to million years back before, there are probably people with more CG. CG is more abundant, but just through all this history, a lot of CG, because of the DNA methylation, this CG pair become a TA pair, and that's today's us. We have less CG than we expected. All right. So this just summarize that the methylated cytosine lead to the mutation because methylated cytosine, when we have lost this amino group, deamination happens high frequency in the cytosine. If this is methylated C, it will turn into the T. If this not methylated C, it will turn to U. And that change can be fixed. But if it's changed to the C, changed to the T, then there's a chance that we will fix it to become an AT, and that's the mutation. So that's that. Now, another important concept about the DNA methylation or cytosine methylation is that we see a lot of this methylation happens in a specific location. this is called a CPG site. So this CPG site is the DNA code. DNA code can be AC, GC, GCAT, TT, etc. And we found that in the location, so for example, I can show you this one here. You probably can see it, but if not, you can get closer, zoom in, you can probably see it too. So this one is the CC, for example, my eye is pretty good, right? CCCG, so this is a sequence of the DNA from a gene to code. This code is APRT. Don't need to worry about what is it, but you can see that this is a long DNA, and you can see that CG are not. If we look at the GC, P is a phosphate, so it's a connection that from the 5' is the initial of the DNA. 3' is the end of DNA. So from the head to the tail, we have CG, that kind of sequence, instead of GC. And we call it CPG because P is the one to connect these two. It's a phosphate backbone. So CG, not GC. Okay, so that sequence means something, right? CG. So if we compare CPG versus GPC, right here, CPG versus GPC, we can see that CPG, that sequence, CG sequence, are disproportionately happens in small range of the DNA code. It happens to here, but not everywhere else. It happens to be the initial segment in the promoter region of the genetic code, but not everything everywhere else. In contrast, it should be, if it randomly appear, it should be randomly appear. It's not only in one location, but not others, right? So this, we call it CPG site, or what we call it CG site or CG island. It's like an isolated region that this is very much like a city, and this very much like a rural area, right? All people are living in the city, very high density. So this makes us wondering why, right? Why they are all located here, but not everywhere else? Because if it's just randomly happening, we should have them to kind of spread everywhere, right? But instead they are in the isolated location. So we call this a CPG site or CPG island, right here, CPG island or CG island. So that's unique. And that's where we see a lot of methylation. And what's the function of the methylation in here? Because now their location is not random. Their location is meaningful. They located in the initial portion of the gene. This is the place we have the Tata box. It doesn't show it here, but this is also the location that control the gene expression. So this is meaningful. And when we have methylation in the CG island, it means suppression. So here is the CG island. So this is just a statistics that so we found this in one gene. This is a APRT gene. And we found that this is not unique. About 60% of this human gene, almost all this housekeeping gene. So those housekeeping gene is not differentiated. These housekeeping gene are the gene to produce normal function of a cell. About all of them have their promoters invaded. with this promoter is the region before the Tata box, right? Before the transcription. And before the location of the initial transcription, right? Has this CPG island. So it's not random. This is spatially located, meaningful location. This CPG island has a length, it's about 300 to 3,000 base pair. So you can get the idea that our gene has long, but our promoter is also long, this promoter segment. And we see a lot of this in this promoter segment. Other portion, we see very less. We call this lower CPG frequency in the rest of the genome. This phenomenon is called CG suppression. We don't see much there. A possibility is the mutation. If we have some there and they don't serve as a function, they probably be mutated into TA, as we talked before. Even the methylation to it, because cytosine is a DNA site that's very easier to get methylation. So the low frequency over there is probably due to the mutation. So that's that. We see a lot only in. So this CPG island meaning the control of gene expression. They are located in the promoter segment of the gene. Now, what's the meaning? Their meaning is to control the turn on or turn off a gene expression. If there is methylation, it means turn off. So here we have some definitive purpose of this DNA methylation. In contrast, the histone methylation is not always turn on or off. So if we look at here, histone methylation. So this DNA, the histone methylation, sorry, not here. So histone methylation can be turned on, can be turned off. This is histone. But if we are talking about the DNA methylation is turned off. DNA methylation is a powerful transcriptional repressor. It turns off the gene expression. So that's the only thing you need to know. That's turned off. And following that, we want to talk about something that is also very interesting. And, just this picture just showing that if it's unmethylated, it's turned on. For example, here we have this is unmethylated CPG island in the promotion part. It leads to the turn on. If we have the methylation in the promotion island, in the CPG island, is turn off the transcription. So methylation on the DNA is turned off. Demethylation is turned on. Okay, so that part you know. That part is easy. Now, the very last portion of this lecture is to talk about the related to the epigenetics. That is that inheritability. That is that this DNA methylation can carry over. Okay, they are the change. They are the modification on the DNA. So the cell with this DNA methylation, when they produce their own daughter cells, when they conduct the mitosis, this DNA methylation can carry over. So you can get the idea that this DNA methylation cause this gene to be suppressed, and that decision can carry over to their own daughter cells. That's right here. The transcription repression of these protein-coded genes is permanent, and then it can affect the all tissue. It means that it can carry over. Let's see this one here. This is called the so here, this is the one. I can mark this one here. We have the DNA. We have the DNA, it's a double-strand DNA. We have the methylation on the C in this 2C, but this 2C is not methylated. So we have the methylated side and unmethylated side. So we have some methylation. This methylation, when they duplicate, one DNA, one chromosome will produce its sister chromatids. The other chromosome carry this CH will produce its sister chromatids. In the beginning, this new copy doesn't have any methylation, but they will produce the methylation. And in the end, two daughter cells will carry the same methylation patterns like their parent cell. So the methylation can carry over during the mitosis. And that's very important, that you need to know that in the beginning, our fertilized cell contains the, this is the zygote. Only one cell contains one pairs of the, one copy of the chromosome from the mother, one copy of the chromosome from the father. This is zygote. In the beginning, there is no methylation, okay? And these cells need to become the stem cell, the original cells, so many different organs. Then they will develop the first heart cell. And when they do that, they need to do some change because they are not the original cell that they can be any cell anymore. They need to do some change. And one of the change is to suppress those potential to become a neuronal cell, to become a basal cell, to become a skin cell, because now they are the heart cell. So that first heart cell has some methylation to it. And that methylation basically defines the fate of that heart cell. And so their daughter cell will carry the same methylation over. So this methylation is stage dependent, stage specific, and tissue specific. If it's brain cell, we don't have the brain cell here, but if it is a brain cell, their methylation gonna be very different from the methylation in the heart cell. And the brain cell, the very first one, has the methylation, and that methylation will carry over to their daughter cells. And so we have a group of cells in the brain. They are methylated. They are methylated because they first pioneer. The precursor cells are methylated the same way that everybody else is methylated. However, their methylation is different from the methylation of the heart cells. So that's that. So DNA methylation is inherited in the mitosis through this somatic cell division. This one is also related to the X chromosome inactivation. That earlier we talked about this sex genetics. That female, she has two S chromosomes, but at some point about during this early cell division, a group of the X chromosome will become inactivated. And that inactivity is due to this methylation. Now we know why it's due to the methylation. And then their daughter cell will carry the same pattern over. And then we have this calico cats, right? That the spot, this region of the skin has the same color of the hair, and the other piece has different color of the fur. And that is because that their X chromosome, the initial one, has this methylation to silent, to suppress one chromosome, one X chromosome. And their daughter cell will carry the same pattern over. So when they divide it, this region will carry the same pattern of DNA methylation, the same pattern of X inactivation. So that's that. So this is the DNA, we call it DNA methylation reprogramming. That is that DNA methylation can carry over the mitosis. However, in the zygote, there's no methylation. So the initial one doesn't have it. So we have, this is a figure showing you what's going on here. These fertilized, this sperm and egg do have the methylation. So this is the level of CpG methylation. This sperm and the egg has the methylation. They have their purpose, so they have some methylation. Methylation basically is to suppress a lot of genes that we don't want them to be functional. Now you are a sperm, just do the sperm work. Don't think about in the past, you probably could be something else, right? Now you are an egg, just do the egg work. So don't think about anything else. Focus on your work, right? You can be like you can be a dancer, you can be a singer, you can be a mother, you can be a teacher, right? And you can be anything. But now you are a student, so you kind of like suppress all other thoughts, just focus on the one. And so every like kind of like functional cells will have a lot of CpG methylation, DNA methylation to it to suppress those like potential. Now, so that's that. We have some methylation. When they fertilized, become a blastocyte, this is the initial couple of the cells, right? Division. At this point, all these methylation are erased. We don't have any methylation. So it's the beginning. And when cells to duplicate, at the point that they are becoming tendency to develop tissue, their methylation is remethylated again. So this is called the reprogramming. And so this reprogramming is stage specific and tissue specific. They are not all the same. Different tissue, different location has different methylation. And then they developed into different organ. One thing that, and this is another stage that when cells produce the conduct this the stem cells for sperm and egg. This is gametogenesis to produce this sperm and egg. This could be the precursor gametose cells. I need to go back and check. But during this gametogenesis, they are all erased because now they are the cells to form the purposed purposed egg cell. So we have two stages that the methylation are erased. One is the gametogenesis, the very first step to produce the in the infant actually, to produce the sperm and eggs, to produce the gamete, and also the stage that's the embryogenesis, that's the beginning of the cell division, of the initiation of a new life. So that's erased. And this methylation can be reprogrammed back to this organism again, and this methylation is stage dependent, also tissue dependent. Stage specific and tissue specific. And they will last for a long time. So that's that. Before the end, I want to talk about another very important concept, that is this pattern of DNA methylation is found to be able to copy from the parents. So the reason is not clear. The mechanism is not fully understood. But it seems like they remember that what kind of DNA methylation happening in their parents. So this is called genomic imprinting, that the expression of the specific gene depends on how their parents express it. So this involves DNA methylation, histone methylation. So that, for example, a well-known gene to be parent dependent is this the gene to encode insulin-like growth factor 2, IGF-2. This is very common, like hormone, to cause the body growth. And although we can carry 2 copies, one from mom, one from dad, but only the one from father is expressed. The one from mother is methylated, is suppressed. So why they choose the mother one to be suppressed, but not the father one? We don't know. And this is also the concept that this methylation, this gene modification is stage dependent and tissue dependent. It affects by stage, right? So it affects by the age and affects by the influence from the environment. So say if you live a very different type of life, maybe you are the first in your generation that you smoke a lot, and that chronic influence will affect your body and to cause a change in your methylation, that you start to modify your DNA histone different way than your earlier generation. Now that methylation patterns has the possibility to carry over to your child, even though that you don't change your DNA. When you smoke, you don't change your DNA, but you do change how you modify your DNA, and that pattern can be carried over. So this is another genomic imprint. So you get the idea that, so this leads to the idea that epigenetics, that our gene expression is affected by the environment, not just by itself. The expression, non-expression is influenced by the environment. A lot of this is related to the tumor, because we know that a lot of our gene actually is suppressed. A lot of our genes have this CpG methylation to it. So even though all cells carry a lot of genes, but a lot of these genes are suppressed. Now, your lifestyle or change may cause demethylation. And the demethylation will make this gene to be expressed. And sometimes that kind of expression can change the cell to become uncontrollably divided, and that becomes the tumor. This DNA or chromatin model remodeling can be carried over to is inheritable. We know that it's inheritable in the cell division, in the mitosis. It can also be memorized in our baby, and it can express the same type. It can have the same type of the methylation pattern in their body. So that's that. So this one is a summary event of this gene remodeling. We basically finish it. In the end, we have some little other things we need to talk about, but the major part we talk about here is the chromatin remodeling. Yes, two types. One is the histone modification and the DNA modification. And DNA modification is on the CPG island or CPG sites. And the DNA methylation is only methylation. Methylation, not like histone has methylation, acetylation, and phosphorylation. DNA has only methylation, and DNA methylation is always suppressed gene, is turning off the gene. All right. So once we have the RNA, DNA to RNA, once we have the RNA, we can conduct some splicing. This part, I think we talked about it already, but since we mentioned about this modification events to control gene expression, I can talk about it again. Kind of like if we talk about it already, this will be just a review, but which is good. A review is always not bad. So this is the RNA splicing. It means that the precursor RNA, the one we first produced the RNA, is not the final RNA. It's immature. It's called a precursor RNA, or we call it a primary transcript. The first draft is not the final draft. It's the first primary transcript after the transcription. It's the one directly produced from the transcription of DNA. Now we need to do some editing to it. And how do we do that editing? The way we do it is called a splicing. So here, before to know the process of the splicing, we need to learn two terms. One is called exon, the other one is called intron. Exon is a segment, exon, intron are placed interleavely. So exon, intron, basically it's like that. And intron is one to be removed. Exon is one to be preserved. So exon will preserve together. And that chopping is like you're cooking, right? You have the celery, and you chop away your root and maybe some leaves, and you keep the one you want. Here, the similar idea. You have this code. You chop away the intron, and then you leave this exon together. All right. So that's this one here. We have the DNA to the RNA. We have the exon, intron, exon. Then we remove this intron and connect all these exons. And we to put some cap to it, put some tail to it. We know that. All right. So that's that. Now, this splicing can lead to some alternative. For example, here is one that we have the exon, intron, exon, intron, exon. We say it exon 1, intron 1, exon 2, intron 2, exon 3, for example. And that's the primary transcript. Now we do this splicing. When we put things together, when we put this exon together, we may only put 2, but not these 2. So we may put exon 1, exon 2, but not exon 3. Or we may put exon 1, exon 3, but not 2. This is controlled by our cell. It depends on the time, the situation, the needs. And we may produce from the same DNA to produce some variety of the RNA. And from this different RNA, we can produce different protein. So that's that. This is the alternative splicing. So that's that. You guys know it. And now that this intron is also, sometimes we call it junk DNA, but nobody call it like that anymore, because we later realized that it's not entirely junk. So it probably has some meaning to it. Multiple protein can be produced. So that's the concept. All right, so that's that. And then this RNA will, so here I want to show you that all these DNA modification, histone modification, and alternative splicing all happens in the nucleus. Only when we have a matured RNA or messenger RNA, it will flow out to become a protein. Okay. So this one just showing you this splicing from the textbook. Exon 1, intron 1, exon A, intron, exon. So we can have A, B, D, or A, C, D, right? Just one code. We can have 4 here, 1, 2, 3, 4, 5 different types of RNA. And that 5 different types of RNA can produce 5 different types of protein. These proteins coming from the same genetic code, these 5 different proteins, we call it isomorph. But it's not mentioned here, so don't need to worry about it. So that just get the idea that same DNA code can produce different proteins through the process called alternative splicing. This alternative splicing is a splice on the RNA process. So that's that. Now, another concept that is very simple is called the microRNA. So basically that now we have this RNA, we need to translate to the protein, and it can be blocked by another RNA called microRNA. So here is the RNA. Now we have this RNA. If it binds with microRNA, they will not be translated. Okay. So just the only thing you need to know about this microRNA is that it silences the RNA. It blocks the RNA. It's not blocking the DNA. It blocks the RNA. So that's that. So microRNA blocks the translation, blocks the RNA. They are small. These are part of this non-coding RNA. They doesn't provide these proteins. They are the RNA only. They are small. And human genome has about 1,000 different microRNA. So the job of this RNA is to make the final stop. That even though we go through all the process to produce this RNA, and they are ready to be translated to become protein, at that moment, microRNA can control their translation. If they bind on it, they cannot be translated. So they can control the speed of the translation. So the amount of protein to be produced can be regulated. This leads to a concept of the technology called the Rna interference. This is the drug that we can duplicate. Say, for example, that the person produced too much of the enzyme, and that can be disease, right? So to control it is that it's already RNA. The way we can do it is because this is in the cell, it's not in the nucleus. And so it's easier that we can provide this drug. This drug is called Rna Interference. It's very similar like the microRNA, and then it will act like microRNA to suppress this RNA translation. So that's that. So we talk about this chromatin remodeling, alternative splicing, microRNA. In the end, we will spend a little bit of time talking about the protein folding. So we know this already. That's why in the very first week, we talk about this large molecule, this organic molecule. The reason is to have the idea about what is the protein. A lot of you guys didn't take the course, didn't take this biochemistry, or you probably did, and you maybe forgot. So we used our very first week to kind of review all these important large molecules. So you have the idea that what is a protein, and the major function, major feature of the protein among those large molecules is that the structure of the protein is very important. So say sugar, like a bread, right? This doesn't really matter the shape of the bread because you eat it anyway, you're gonna digest it. But the shape of the glove is important, right? Because glove is one from the like fiber, and you knit it to become a glove. And that shape is important. If it's not the same shape, it won't be a glove, right? Or sweater, right? You knit it to make a sweater. If it's not this shape, it won't be a sweater. And protein is like that. Protein has a structure, and the structure is because of the knitting to make it a shape like that. And that's called the protein folding. So here is, we use an example of this DSPP gene. This is a special like gene to code the protein. And so we have the DNA transcribed to RNA, transcribed to protein. Okay, so everything's fine here. Now, if we fold it in one way, and we can fold it in another way, different folding will make the function of the protein differently. And in this case, it will be degraded slower. This is more common. This is the functional one. Or degraded faster. This is rare, and this is no. So folding is important. I think that's it. So this one summarizes it, that I also put a number here. The number of the gene is not like strikingly large. The number of gene is not that much, but from the same gene through a lot of control, we can have a larger number of the messenger RNA. We can have some present, some express, some modification. We can have some alternative splicing, and so the same code can have multiple different type of the messenger RNA products. And the same messenger RNA products can have multiple structurally different protein because we can have some modification, different folding. We can have different decoration too. We can have phosphorylation to it. We can have glucose add to it. It's called glycoprotein, different decoration. So same gene, we don't need to have a lot of gene. We can still have a very rich, diverse protein in our body. So that's that. Here is two last pages. It's about this key concept. So this chromatin remodeling, two type remodeling, histone remodeling, and the DNA remodeling. Modification, sorry, the histone modification and DNA modification. Histone modification has three types: acetylation, methylation, and phosphorylation. The DNA modification has only one type, it's methylation, and the methylation is in the CpG site, and the function is to suppress gene expression. So that's that. As the acetylation, phosphorylation, methylation, all has this function. I don't. Acetylation, sorry. So just. Acetylation on the methylation. Acetylation and the phosphorylation. We didn't really talk about the phosphorylation, but you just need to know that acetylation. So if you don't know phosphorylation, it's okay. Acetylation can turn on transcription. It's DNA methyl group. So it's especially on the CpG island can turn it off. So acetylation turn on a gene. Methylation in a CpG turn it off. The patterns of the pattern of the so the DNA methylation is heritable. That we know that it can carry from cell through the mitosis to the daughter cells, and there is also possibility that from the methylation pattern from the parents can carry over to their offspring. Although the mechanism is still not fully understood, but we do have that kind of observation. The next one is the microRNA can silence the RNA translation. And the last one, but not least, is the that's how the number of the gene is limited, but we can produce so many different type of the protein that is due to so many different steps of the regulation or control of this gene expression. Alternative splicing and the cutting of the precursor protein maximize the number of protein that DNA can encode it. So that's that. Hope you kind of gain some more knowledge about this gene expression and very importantly that even though you cannot control your DNA, the coding, you cannot control the sequence of the DNA, like that's from your parents, right? But we can influence how we express it. These are epigenetic, the term that you commonly hear from the news or from the media, refer to all these modification, especially the chromatin modification, this histone modification, DNA modification, that is influenced by the environment. So you can make a change, because even though you carry the same gene like your parents, you have the same gene like your brothers, sisters, you can be different. You can live a different life. This is not just encouraging like lecture. This is physiology. This is human genetics. This has science to support it. So from now on, it's never too late. You know, start to live a self-disciplined life that it will. Don't hang out with people that has bad influence, because they bad influence not just you, but it will influence your own gene, and that modification can influence your kids. So a healthy, live a self-disciplined life, sleep well. When the more you mess up with the body, the chance you will one day you will not be able to handle it. All right, so that's that. Thank you guys. See you later. Let me know if you have any question, okay?